السلام عليكم ورحمة الله وبركاته. بسم الله الرحمن الرحيم. This is the third lecture of session 9 about testing hypotheses. In the first two lectures, we went through two examples of how to assess the quality or the goodness of a model. Inshallah, this lecture will go through the actual framework of testing hypotheses. We will define the terminology and we will apply it on a third example. Let's start by a summary of what we have done so far. So this is the method of how we can measure the quality of a model. So we have a categorical sample, and there is an assumption about the categorical sample that it is drawn randomly from a known categorical distribution. So that's the model that we want to test its quality. To do that, we have to decide on a statistic that will help us decide whether this model is good or bad. So usually we pick up a statistic that measures, in this case when we have a categorical sample drawn from a categorical distribution, we pick up a statistic that measures the distance between the two distributions. The one that we see in the data and the population, which is the assumption of the model. We compute the statistic from the sample and we call that the observed statistic or the observed value of the statistic. And we also simulate the distribution of the statistic from the population. So we sample at random from the population. For each sample, for one sample, we compute the statistic and we repeat that multiple times. And then we compare two things. We compare the empirical distribution of the simulated statistic because we simulated the statistic here by randomly sampling from the population and computing the statistics. Now we have an empirical distribution of the statistic like the one in the figure here. So we compare between this distribution and the observed statistic that we already computed from the data that we have. So we start by a sample of the data. We have an assumption that this data was randomly drawn from a population. So to test this assumption, we will simulate getting samples from the population. We look at the distribution of these samples, actually the distribution of the statistic computed from these samples, and we compare this distribution with the observed statistic. So that's what we have done. We applied it on two examples. The first one was the coronavirus example, and the second one was the jury selection. Let's now put all of this into a framework and we see what is the terminology that we should use. So first, let's say that testing hypotheses is about choosing one of two viewpoints of the world. The first one is the model that we have. For example, the people on the jury panels were selected at random from the eligible population. That's the example from the second application we had in the previous lecture. So that's an assumption. We assume that people of the selected people from in the panel were randomly selected from the eligible population. That's one view of the world. The other one, the alternative viewpoint, is that no, they were not selected at random. There might be some other factors that affected the way the panel was selected. So these are the two viewpoints. So usually in testing hypotheses, we have two viewpoints. One, we build the model upon assumption. Here, the assumption is that the panel was selected at random from the eligible population. And the other alternative, the alternative viewpoint is that no, it is not. So more formally, hypothesis testing is a test that helps us decide between two views of how data were generated, which are the two viewpoints we talked about in the previous slide. The two viewpoints are called hypotheses. Each one is a hypothesis. The two views or the two viewpoints are called hypotheses. So in the jury selection example, the two hypotheses were the jury was selected at random, the other one it's not. And the test should eventually pick the hypothesis that is better supported by the observed data. Remember, we always have observed data. We always have a real sample from a real sample that we want to test whether it came from a specific model or not. So from this real sample, from this real data, from this observed data, we want to see whether it fits the model or not. Okay, so the test should eventually make us or helps us decide which viewpoint is more supported, is better supported by the observed data. So we said we have two hypotheses. The first one is called the null hypothesis. The null hypothesis. And in the null hypothesis, we assume that the data are generated by a well-defined chance model or a well-defined random process. Okay, so there is an assumption here that the data is generated randomly by some random process or by a chance model. If that's the case, then we can simulate the data under that assumption because it is well-known, well-defined chance model, well-defined random process. We can simulate getting data under that assumption. We call that simulation under the null hypothesis. Now, why do we call this hypothesis the null hypothesis? We call it that way because we assume that there is nothing affecting the sample that we get or nothing about generating the data except chance. Nothing is affecting the generation of the data except chance, except randomness. Okay, so nothing more is more than chance. That's why we call this the null hypothesis. The other one, unsurprisingly, is called the alternative hypothesis. In the alternative hypothesis, we assume that there is some effect other than chance. So in the null hypothesis, we said that everything is by chance, everything is random, everything is under well-defined random process or chance model. In the alternative hypothesis, we say no, there is something else going on other than chance. So this is the different view about the origin of the data. In the null hypothesis, we assume that the data was generated by a well-defined process, a random process. In the alternative hypothesis, we assume that there is something else that we might not know affected the way that data was generated. The method, the hypothesis testing, only works if we can simulate data under one of these two hypotheses. Because we want to test, right? So we have to simulate some data, okay, from one of these two hypotheses and compare it with the sample that we have. Which one of these two hypotheses can be the hypothesis by which we can simulate or for which we can simulate data? Of course, the null hypothesis, because we assume that it is it is affected by a well-defined chance model or random process. Okay, so we will always simulate data under the null hypothesis, not under the alternative hypothesis. So remember always that the null hypothesis is all about randomness, and that's the hypothesis that we will simulate data from or about. The other one, there's something else other than chance. Maybe some chance, but maybe something else. Okay, so the alternative hypothesis is all about something going on that we might not know in addition or in absence of chance or randomness. But the null hypothesis is very clear. It's all about chance model or random process. Now, to do the test, we have to choose this statistic, or more formally, the test statistic. So the test statistic is a statistic that we choose to simulate in order to decide between the two hypotheses or the two viewpoints. Now, to choose the statistic, there are some questions that we have to answer. Actually, two questions. The first, what values of the statistic will make us lean towards the null hypothesis? In other words, what values of the statistic will make us decide that the null hypothesis is good? On the other hand, the other question is, what values will make us lean towards the alternative hypothesis? So what are the values of the statistic that will make us decide that, okay, no, the null hypothesis doesn't look good, is not good, so we will reject it. So it's an evidence against the null hypothesis, or as we said it before, it's an evidence against the model, the random model, or the chance model. Okay? When we select the statistic, there are many ways to select a statistic for a specific problem. But we always prefer the statistic that will be just high or low to lean towards the alternative. We don't prefer, or we try to avoid, the values that will be high and low to indicate that the alternative is good or the model is bad. Okay, so we always prefer that when we pick the statistic, when the statistic is high, that means that this is evidence against the model, or low. So one side of these two extremes. Okay, either high means the alternative, or low means the alternative. We don't prefer the statistic that if it's too small, it is supporting the alternative, or high, it will support the alternative. We just prefer one of these two sides. We'll talk about that more today and in the next lecture also, inshallah. Now, let's assume that we picked the statistic. We can now predict its values under the null hypothesis. Why under the null hypothesis? Because we know very clearly the process of the null hypothesis. The process in the null hypothesis, we know clearly the process behind generating the data. So we can simulate this generation process, and we see what values will be predicted for that statistic. So we simulate the statistic under the null hypothesis, and we draw the histogram of the simulated values. This will show us, the histogram will show us the empirical distribution of the statistic under the null hypothesis. So it is a prediction about the statistic. What are the possible values that we can see for the statistic by randomly sampling from the population? Randomly sampling according to the assumptions of the null hypothesis, the assumption of the random or the chance model. So it shows all the likely values of the statistic, but not only that, it also shows how likely each of these values will be if the null hypothesis is true. So when we do the simulation, we actually assume that the null hypothesis is true. And that's how we generate our samples. So we assume that the null hypothesis is true. We get a sample based on the well-known random process. We compute the statistic. We repeat that multiple times. What is this process doing actually? We're trying to predict what possible values of the statistic we will get from random samples from the population. Remember that these probabilities, so we said that the histogram will show us two things. Will show us the likely values of the statistic and the probability of each one. But these probabilities are approximate. Why is that? These probabilities are not exact. Of course they are not exact, just because we cannot generate all the possible random samples to get the exact probabilities. Getting exact probabilities means that we have to generate all the possible random samples from the population in order to get the exact values of the probabilities. Of course, we don't have time and resources for that. We just simulated for some good number of samples, and that's why we are saying probabilities are just approximations. And that's why we call it an empirical distribution. Okay. Now, after we get the histogram, we need to do a conclusion. We need to make a decision. So the decision is all about the conclusion of the test. So we have to choose between the null hypothesis and the alternative hypothesis. So we compare the observed test statistic that we computed from where? From the data sample, from the sample that we have, not from the sample that we simulate. Okay, so the observed sample is the data that we have. We compute from that sample the observed test statistic. That's the reality. That's the real value that we see. Okay, so we compare this real value with the empirical distribution under the null hypothesis. If the observed value is not consistent, or if it's inconsistent with the distribution, that's now evidence against the model, or we can say that now the test favors the alternative. Formally or statistically, we say we reject the null hypothesis. So the null hypothesis is not good. We reject the null hypothesis if the observed value is not consistent, if the observed value of the statistic is not consistent with the empirical distribution of the statistic. Now, to decide whether the value is consistent or not, we can do visualization as we did before. Sometimes the visualization is sufficient, or we can have some conventions about consistency, and that's what we will discuss inshallah within the example that we will go through just right now. Okay, so that's the framework of hypothesis testing. We have two viewpoints of the world. We call them two hypotheses. One is the null hypothesis, which is all about randomness. The other is the alternative hypothesis, which is indicating that there's something else going on other than chance. We pick a test statistic, we simulate the values from values of this test statistic from the population. We compare the empirical histogram of the statistic with the observed value, and we make a decision based on that. Now let's apply this on a nice example, nice application about genetic models. So I think many, or if not all of us, are familiar with Gregor Mendel. He is a scientist who has many contributions in the genetic field, and that's why Google actually acknowledged his contributions by a very nice doodle which is shown here. One of the models that Mendel has is about pea plants, or a particular type of pea plants. So he noticed that the flowers of this plant has either purple flowers or white flowers, and he had a model in mind. So the model was that each plant is purple flowering with a chance of 75%, which means that 25% will be white flowering. So that's the model that Mendel has. Now, to test that model, Mendel grew some of this type of plants, and he observed how many of them are purple flowering and how many of them are white flowering. So that the model was regardless of the colors of the other plants. So it's 75%, 25%, and that's it. That's the random, the chance model here. So the two viewpoints that that we have are the first one is that the data that Mendel collected is generated from that distribution. Okay, which is 75% purple, 25% white. The other one, it's not. So there's maybe some other model or something else other than just chance by that distribution that affected the color of the plant. So the question is this model good or bad? So the two hypotheses here, the null hypothesis is that Mendel's model is good. Mendel's model which is 75% purple, 25% white, is good, and the alternative hypothesis will be no, it's not. So how can we test that? Let's first choose a statistic. So what kind of statistic we should use here to help us make a decision whether Mendel's model is good or bad? So let's think together. We have a population based on the model, based on the null hypothesis, which is 75% purple and 25% white. So maybe we should look at from any sample, we should look at what percentage of it is purple. Okay, now if that percentage is larger, is bigger than 75%, is much bigger than 75%, then this is evidence against Mendel's model. So for example, if it is 95%, so might be evidence against the model. On the other side, if it is much less than 75%, let's say 10%, then it is also evidence against the model. So this is the case where if we chose that statistic, low values and high values both will indicate, will make us lean towards the alternative. So it's better to make it one-sided. Okay, so we can say that this statistic is the distance between the percentage that we see from the sample and 75%. Now that distance, of course, the absolute value of that distance will always be high to indicate that to make us lean towards the alternative, or to be evidence against the null hypothesis. Okay, so we can start by the percentage, but as we said, much larger, much smaller than 75 will be against the model. So the distance from 75 is the key. So the statistic that we should use here, or preferred here, is the sample percent of purple flowering minus 75. Again, there are different ways of selecting the statistic. So you could also select the statistic that is just this part without the minus, without the subtraction. But in this case, you have to check if it's too low or too high. But if we just do the distance, which is just here the difference, it will be just one side that we need to check, which is if it's too high or not. Okay? Or if it's too far from 75 or not, which is the meaning of this statistic. So if the statistic is large, this is evidence against the model. Okay, so now that we chose a statistic, what is the next step? What should we do now? We should do a simulation, right? We need to compare the value of the statistic that we can compute from the sample that Mendel got with predicted values of the statistic that we can compute from random samples through simulation. So let's do that together. So here is the Mendel experiment. This is the model proportions. So we have 75%, 25%. Now let's compute. So Mendel had 929 plants. So that's the sample, that's the real sample, 929 plants, of which 709 had purple flowers. Okay? So the observed percentage of purples is 709 over 929. That's the percentage of the purple flowers in the real sample, in the real data. Okay? And this is about 67.3%. Now, this is not the statistic that we agreed upon. The statistic is the absolute difference between this value, so that's the observed statistic, the absolute difference between this value and 75%, which is 0.013. Okay, so that's the observed value of the statistic. Now, let me ask you a question and think about it. Is that value high or low? 0.01318. You might be tempted to say, okay, this is low. Actually, we should not say whether it's high or low now. We cannot, because we don't have a reference. We have to compare it with the empirical distribution so that we can say it is high or low. So don't fall into this trick. You cannot, just from the observed value of the statistic, say I should reject the null hypothesis or not. Wait until you do the simulation and you get the empirical distribution. Okay, so we cannot make any decision looking at this value, whether it is 0.013 or even 0.95. Okay, so we have to look at the empirical distribution before we make a decision. So let's continue in the simulation. This is our friend sampling function that we used in the previous lecture, previous two lectures actually, sample proportions. So the sample proportions function takes two arguments, the sample size and the distribution of the population. So the distribution of the population here is Mendel's assumed distribution, which is 75% and 25%. The sample size here has to match the size of the real sample, the size of the data sample, which is 929. Okay, now let's do that once and see what proportions we get. So here we got 73% and 26%. So we can repeat it multiple times, and instead of that, let's put that into a function that will give us the value of statistic of one sample. So we said that the statistic will be the absolute difference between the percentage of the purple flowering, which is the first item of the returned distribution from sample proportions, minus 0.75. Okay, so that's the function that will give us one value of statistic. Now we can call that function. So here is one value, which is 0.015. If we call it another time, 0.18, third time 0.21, and so on. So we need to repeat that multiple times. Okay, here I will repeat it. So let's make it less than that, 10,000 times. Okay, and from in each repetition we will get one value of the statistic, which is the new purple here, and we append it to an array called purples. So this is the process that we did so many times before to collect the values of the statistic before we show it in a histogram. So if we run that, now we have 10,000 values, and let me now show the histogram. So here is the histogram. Okay, so that's the distance from 0.75, and these are the predicted values of the statistic. Okay, so that's the empirical distribution. That's the empirical distribution. Now, what is the next step? We got the empirical distribution. What should we do next? We should compare it with the observed value of the statistic. Where is the observed value? This one, which is the observed statistic. Okay, so to make the comparison easier, we will just plot this value as a red dot here. Okay, so the red dot here is the value of the observed statistic. Okay, so now we have both on the same plot. We have the empirical distribution and we have the value of the statistic. Now we can answer the question of whether this observed value is small or is low or high. Okay, and more importantly, we will answer the question whether whether the data and the null hypothesis are consistent. The data and the empirical distribution are consistent. So what do you think? Is it consistent or not? Now, looking at the observed value of the statistic and the empirical distribution, it seems that the observed value lies at the heart of the distribution. So, to me, it looks very likely that the sample data came from this distribution, because this value of the statistic in particular is not very unlikely. It's not very rare value. It appeared a lot in the distribution, in the empirical distribution or the simulated data. So that's evidence towards the null hypothesis, and we can now say that we cannot reject the null hypothesis. Okay, so the decision is the Mendel's model is good. Okay, now, so it might be quite clear that this is consistent with the model. But what if this value is here or here? So we'll start to be more worried when we move this value to the right, right? Getting the value here, it's less likely. There's a value here, it's very unlikely. So where should we say that, or at what time, at what exact value we should say that the model and the data are inconsistent? So maybe here it's consistent, here consistent, here consistent, here consistent, here quite can say, okay, consistent. Here we are more worried, here we are even more worried, and so on. So when can we have a cutoff? When can we say, okay, now we can say it is consistent, or now we can say it is inconsistent? So the answer to this question is that it is a subjective matter. What do you mean by subjective matter? It is a matter of your judgment. So if the data is here, if the observed statistic is here, one can say it looks to me consistent with the distribution. Another one can say no, it doesn't look consistent, it looks inconsistent. So we might have different opinion. So that's why we call it is subjective. Of course, we all, or most of us, like to have rules, like to have a rule that says, okay, look at this value, do some calculation, and based on that calculation, we say it is consistent or it is inconsistent. So let's try to come up with a rule, if any, that can help us make this decision easily. So let's look at the simulated samples, and let's see what percentage of the samples gave us a value of the predicted or a predicted value of the statistic higher than this value. Okay, how can we do that? So if we look at the distribution to the right of the observed value, and we look at the area of the distribution to the right of this value. So what will be the area? What is the meaning of the area to the right of this value? Remember from the histogram, what is the meaning of the area? So the area in histogram represents percentage. In our case, it will be a probability, right? So percentage of the values that are above this value. So that's if we look at the area of the distribution to the right of this value. Okay, how can we compute that? We know how many, so we know we get all the values that are the sampled values. We can see how many of them are above this value, divide by the number of samples, which is the repetitions. So this is this is one way of doing it. So purples is the array of the sampled values. We want to get all the values that are, or the number of values that are at or above the observed statistic, and we divide that by the repetitions, we get the percentage. So if we do that, we will get 35 or 36%. So 36% is above this value. So we might say, I think we can agree that this is high enough values that are above that statistic, so we can say this above this observed value. So we can say that this this value is consistent with the data. Now let's try another one that might be problematic. So let's pick something maybe here, okay, and see what this value will be. So something like 0.035, something between 0.003 and 0.04, so 0.035. So let's take this, maybe copy it and change this value to be 0.035. Okay, now when we do that, we got this value, 0.01, sorry, 0.01, which means that 1% and a bit of the samples gave us a value that is 0.035 or more. Okay, so the percentage of the tail at that value is like 1% or a bit more. So you might look at this value and say, okay, to me, that looks a very low value, 1%. So I will say this data, this observed value is inconsistent with the model, and I will reject the null hypothesis. Okay? Okay, now let's do that again with another value. Let's say 0.03. That will give us 0.0347. Now it's 3% or more. Let's do it at 0.025. It's 8% now. Okay, so you look at these values and you make your own threshold. So someone might say, okay, if this value is, let's say, 10% or less, I will say it's inconsistent. Someone else might say, okay, no, my threshold is 5%. My threshold is 6%. So it's subjective. But at least now we have a value that we can check and say if it's above, if it's below my own cutoff point, then it is inconsistent. Now, statisticians and, as I said, even practitioners don't like that. They like to have some set rule, okay, some cutoff. So there are in the field some conventional cutoffs that came from somewhere. We'll talk about that inshallah in the next lecture, but there are conventional cutoffs. Actually, we have two cutoffs. The first one is 5%. So if this value is less than or equal to 5.05, which means 5% of the sampled statistic values are at or below the observed statistic, they would say that's inconsistent. Okay, and they call this value small. So if the value of if that value is less than 5%, they call it very small. They call it small. If it's less than 1%, they call it very small. So these are two conventional cutoffs that are used all over the place, 5% and 1%. So if my observed value gave me a value of less than 5%, that we can call the result statistically significant. If this value is less than 1%, we call it highly statistically significant. So these are two conventional rules that are frequently used, but they are not golden rules. They are not the only rules. Usually we use them, but as I said, originally it is very subjective. And because it is very subjective, when we have something like this in data science, we usually report, or we'll have to report, two things. We have to show the distribution and report this value. Okay? We report the value which is the tail probability. So we call it tail probability. Okay? Because that's the value, that's the probability that the statistic will be at or higher than, in this example, at or higher than the observed statistic value. Okay, so we show the distribution, the empirical distribution, and we also report the tail probability. Okay, so that's the end of this example. In the next lecture, we will call this value, we give it a specific name, and we'll go through more examples and show how we will use these conventions and how we will use this tail probability value. Okay, inshallah we'll see you in, or actually talk to you very soon for the next lecture, which inshallah hopefully will be the last lecture in the session of hypothesis testing. Talk to you or see you soon inshallah. Assalamu alaikum wa rahmatullahi wa barakatuh.